Okay, good morning, ladies and gentlemen. I am Belle Shenoy, Managing Editor of MycoAsia Journal of Modern Mycology. Today we have with us Professor Samantha Karunarathna, who is also a Managing Editor of MycoAsia Journal of Modern Mycology. Sam is a very familiar face in mycology, especially when it comes to Basidiomycetes. A little bit about Sam. Sam is Sri Lankan, but currently based in China. He is a professor at the Center for Yunnan Plateau Biological Resources Protection and Utilization at Qujing Normal University, Yunnan, China. He holds an adjunct professor position at the National Institute of Fundamental Studies, NIFS, in Sri Lanka. He completed his Ph.D. at Mae Fah Luang University in Thailand and his B.Sc at the University of Peradeniya in Sri Lanka. His work primarily focuses on taxonomy, phylogeny, and domestication of fungi, especially Ascomycetes and Basidiomycetes. He has published extensively on wild animal, medicinal, and luminous mushrooms. He has co-founded MycoAsia in 2019. He is the editor-in-chief of our new fungal taxonomy journal, Fungal Taxa, Systematics and Evolution. With this brief introduction, I invite Professor Sam to deliver his talk on how to identify mushrooms to species level. Over to you, Dr. Sam, and thank you very much for accepting the opportunity.
Yeah, thank you very much, Dr. Shenoy, for inviting me to deliver this talk. I think we postponed the talk a couple of times, but finally, I'm happy that I can do the talk today. So let me share my presentation. Okay, can you see my PPT?
Yes, I can see you, but can you drag that the small window aside? Okay, great. Thank you. Yeah, thank you.
Okay. Good afternoon to you all. So my topic today is mushroom identification. So I'm not going into the molecular aspects because it is a separate talk. So I'm talking about the macro and micro morphology associated with mushroom identification. I believe this is very important in mushroom identification. So mushrooms, as you all know, they have different shapes, different colors, and they are the above ground fruiting bodies of fungi, and they produce reproductive spores in the fruiting bodies. So when you go into the field to collect mushrooms, you need these things. So you need a knife in case if you want to pull out some mushrooms from dead wood. So plastic boxes to keep the mushrooms safe, or baskets, aluminum foil to wrap the mushrooms, and a good camera. These days we use smartphones, it's easy, and GPS to record the location, and boots, especially when you do field work in tropical areas, you know, the leeches and snakes, and raincoats, because we collect mushrooms in the rainy season, and leech socks to prevent leeches, and a first aid box because we don't know what things we face when we are in the field. So these are the habitats of mushrooms. So mushrooms grow on pastures and soil, leaf debris, rotting wood, and even the live wood. So when and where to find mushrooms? As I said, mushrooms pop up when it rains. So in tropical areas, mostly it is May to September. And normally they grow on dead or dying trees or soil. And mushrooms like moisture. So if there is no rain, you don't see mushrooms pop up. And these are some tips for mushroom hunting. So some mushrooms, they are associated with certain trees, like we call ectomycorrhizal mushrooms. So they are associated with ectomycorrhizal trees, like Russula lactarius. So they grow associated with these ectomycorrhizal trees. For example, pine and Castonopsis, these kind of trees. And mushrooms also tend to grow in unsettled areas. For example, burned sites, and also the areas disturbed by floods. So, and also logging areas, and all the areas that are disturbed by humans. So in mushroom identification, taking a good photograph is very important. So you can spend some time and take a really good photograph that will be important in identifying the mushroom. If you can take photos from the above, from the below, these photographs can show all the characteristics of the mushrooms. So you can see these are some of the photographs of mushroom photographers, how they take good photographs in the field. So also keep in mind to keep something with the mushroom to know the size of the mushroom, whether it is a coin or it is a knot. You can put there and take the photograph, so you can see the size later. No need to worry about that you forgot to measure the mushroom. And you can see this is one of the photographs which was taken by Steve Axford, one of the best mushroom photographers. So you can see the details in the stipe, in the cap, and the vulva. You can see it is very detailed photograph. Nowadays, we have the smartphones. The cameras are really good. You can take these kind of photographs. And this is Cordyceps militaris, also by Steve Axford. You can see the tiny details in the photo. And as I mentioned earlier, you should take the photographs in order to show the above and below characteristics. So these things are very important in identifying the mushrooms. So this particular mushroom is Agaricus blazei or Agaricus subrufescens. So the ring below has a very characteristic features. You can see here, like the cottony appearance in the ring before it opens. So these things are very important in identifying mushrooms. So you can see here also, this is a Lepiota species, that the above and below and the ring and everything is very clear. So when collect mushrooms, so dig around the stipe, stipe base, and if there is a vulva or bulb, a root, or insect fungi, sometimes the insect is inside. So you need to dig out all these things in order to get the full picture of the mushroom. So the substrate also very important. Later you can see what kind of substrate that this mushroom was growing on. So also it is important to record all the associated plants around. So as I mentioned, the ectomycorrhizal mushrooms, they are associated with certain trees, and also take several sporocarps or the basidiocarps, and showing different range of development. Sometimes it's the bud stage and a little bit open and fully opened. All these stages are important. And when you find one mushroom, so can observe the surrounding areas, so you may find more specimens. I'm telling to find more specimens because, when you describe a new species, single specimen is not enough to describe a new species. So it is always important you find two collections in order to properly describe a new species. I'm not telling that one specimen cannot describe a new species, but it's very hard for you to publish this kind of new species because most of the journals, they require two collections. And so after you collect the mushroom, you need to store them properly, as I mentioned, in plastic boxes, and you can wrap in aluminum foil to avoid the losing the moisture. Otherwise, the mushroom will go bad quickly. And you have to keep the collection separately. Do not mix the collections. If you mix, then later you have contamination problems when you sequence your specimens. So, as I mentioned, the macro characteristics of mushrooms are very important when identifying mushrooms. So, depending on the different groups, so mushrooms are categorized like this in gilled mushrooms, which contain gills, and cantharellus, you can see they have gill-like structure but not gills. And tooth fungi, they have tooth below the fruiting body. And the club or coral fungi, it looks like a coral. And jelly fungi looks like a jelly. Crust, you can see most of the time growing on dead wood. And bolets, you can see the pores. And polyporales also have big pores in the fruiting body. And bird nest looks like a bird's nest because the spores inside, you can see it's like bird's eggs in a bird nest. And stinkhorns, they have these sticky spore mass. This is an adaptation to disperse their spores. And puffballs, and earth stars, and morels, cup fungi, and the lice. So these are the major parts of a mushroom. So when you collect a mushroom, you need to see whether the mushroom contains these parts. Like not all the mushrooms contain all these parts, but at least one of these parts should be there in the mushroom. So this one we call pileus or cap, and the parts that remain on the cap we call universal veil remnants, because when the mushroom is small, the mushroom is covered by this universal veil. So when the mushroom opens up, some parts of the universal veil remain on the cap. So so we can see like scales, and the other part remain as partial veil or the ring, and the other part remain as vulva sometimes in the mushroom. So this below part is the fertile part, that contains the basidiospores, so we call hymenium, and the stipe and mycelial threads. So when you collect a mushroom, it is important you collect all these parts. Otherwise, if you collect a mushroom that has a vulva, but when you collect it, if you did not collect the vulva, then when you describe the mushroom, it gives wrong information. And when you describe mushrooms, so we need to check all these characteristics that we have different forms that include all these characteristics. So gill attachment is also important when identifying mushrooms. So some mushrooms, the gill attachment is free, some adnate, some adnex. So we have different terms for these. And cap shape, ovoid, globose, such as we have a lot of different shapes. And stipe shape, we have different terms for these. So when we describe a mushroom, we need to include all these terms. And the margin of the cap also different. So we need to describe the term according to the mushroom. So as I mentioned, these are the important things that we should record when we collect a mushroom. So date, it is very important that you collect, and the location. If you have a GPS, so you can write down the GPS location, and the habitat, what kind of habitat, and what kind of trees, and the substrate, whether it is a dead wood, whether it is a live wood, or soil, or leaf litter, and the habit. And so these things are very important. And also for agarics, bolids, these characteristics are important. The cap, color, shape, size, texture, as I mentioned, the margin and the veil, and then the gills, the details, pores, and the stem, spore print color. I will tell later how to take spore print, and any other distinguishing features that you see. So basically, you need to write down or note down all the characteristics that you can see when you describe the macro characteristics. So when you study the characteristic on fresh specimens, as I mentioned, these are the things you need to note down. And smell and taste. So, you know, some mushrooms like Amanita, so they are known for toxic, their toxicity. So some Amanita can be deadly poisonous. So in this case, I do not suggest you to do the taste. We know sometimes we take a small piece and then we use our the tip of the tongue to check the taste of this mushroom. Sometimes some Rashulas or the Lactarius species, you can feel the burning sensation when you put it on your tongue. So soon after you taste it, you spit it out and you can wash your mouth. But regarding Amanita, I don't suggest to do the taste because we never know whether it is a deadly poisonous one or a normal mushroom. So for non-genera like Lentinus, Pleurotus, like these kind of mushrooms, it is okay to do the taste, but for Amanita, I do not suggest. And as I said earlier, so you can see this is the ring of Amanita rubrovolvata. You can see it is very clear in this photograph. And here you can see the partial veil. I told you that sometimes it remained on the cap as the scales. And this is Agaricus tricepheratus. So you can see the universal veil remnants on the cap and also on the stipe. And this is the clear picture of a volva in Amanita hemibafa when it is very young. So when we talk about Basidiomycetes, the characteristic is this septum, the dolipo septum, and also the mycelium is usually dikaryotic, and also not all are mycelial. Yeast, some Basidiomycete yeast, they are cellular, like single cells. And clamp connection is also very important when we talk about Basidiomycetes. So in the vegetative hyphae, you can see this kind of structure we call clamp connection. So sometimes when you find asexual stages of Basidiomycete, so it is hard to know whether this is a Basidiomycete or not. But when you observe under the microscope, if you see these clamp connections, so you are certain that it is a Basidiomycete. And usually they produce 4 basidiospores, but sometimes you can see 3, 2, or sometimes 1. So when you study mushrooms, this is the sectioning technique. So usually you cut a piece from the cap like this, and then you take a section in this direction to observe the gill surface and the hymenium. So in the hymenium, you can see these structures. So when you study the micro characteristics, it is very important you section in this way and see all these parts. So we call this part is pellis and hypodermium, the below cell surface, and paliostroma, and then subhymenium, and then this is the hymenium surface. You can see all the different cells. So depending on the place you find the cystidium, you call these cystidia in different names. So if you see cystidia in the tip of the gills, so you call it chelocystidia. And if you see the cystidia in the side of the gills, you call it pleurocystidia. And then you see the basidia that contains basidiospores, and the young basidia we call basidioles. And so you may wonder what is the purpose of having these cystidia. The cystidia are bigger cells than normal basidia. So because of these bigger cells, these gills can keep apart from each other. Because you can imagine if there are no cystidia, then the gills touch each other. So this is not good for spore dispersal. Because if there is a space in between, so it is very easy for mushrooms to disperse their spores. So this is the basic function of having bigger cells in the gills. So these are the bigger picture of the Basidia and Cystidia. You can see in here. So it keeps two gills apart, and there is a space in between. So it is easy for spores to disperse. So when we talk about the basidiospores, there are different shapes. So this is a globose, ellipsoid, oblong, and nodulose, and cylindric, fusiform. So these shapes are also important when identifying mushrooms. And ornamentation of basidiospores, also important. Some basidiospores are smooth, some are verted, and some has spines, spiny, and then reticulate and striate. So as I mentioned earlier, like the spore print is very important. So this is how you do a spore print. So I suggest to do a spore print by using one black and one white paper. Keep like this, and then you put the cap of the mushroom on the top of the papers, one half in the black, one half in the white paper. So if the spore print is darker one, you can see it on the white paper. If the spore print is a light one or white color, you can see it on the darker paper. So you can use this method, then you will not have problem whether it is a dark spore print or light spore print. So Amanita normally has white, Volvarella pink, Chlorophyllum olive green, Agaricus chocolate brown. So basidiospore color is important in identifying, as I mentioned. So white spore print can see in these families and orders, and pink in these Entelomaceae and Proteaceae, and purple brown, rusty yellow brown, earth brown, brown, black. So the spore color is important. So that's why I mentioned that it is important to do a spore print to know the color in advance. So here are some of the examples. You can see their colors and also the shapes, different shapes. So when you see these kind of spores under the microscope, it's very beautiful because they have different ornamentation and different colors. And so Basidia, as I mentioned, usually it is 4, but sometimes 3, 2, or 1. And this is how you can see the basidiospores and Basidia under the microscope or SEM. And these are different types of Basidia. So depending on different groups, the shapes and the structures of Basidia are also different. So we call this one is holobasidium, single-celled, and you can mostly see 4 basidiospores. And these are heterobasidium. You can see in jelly or rust fungi, they have multi-celled and different structures. So cystidia, I told earlier what is the purpose and how we call them according to the different the location of the cystidia. So if it is in Pileus, we call Pileocystidia. It is in lamella sites, we call Pyrocystidia. If it is in the edges, called Chelocystidia. If it is in stipe, we call Colocystidia. So depending on the place, we have different names. And also the morphology is different. Shape, size, ornamentation, color can be different. So these are different shapes of cystidia. So depending on the shape, we have different terms. So we need to mention these terms when you describe a mushroom microscopically. So after we combine both macro characteristics and micro characteristics, so we have a full description of the mushroom. So this full description is very important. Nowadays we see like some people describe fungi based only on phylogeny, which is not, in my opinion, which is not correct. So we always should consider the morphology in order to describe it properly. Otherwise, how can you imagine a mushroom by looking at only a sequence? But when you describe a mushroom based on macro and micro morphology, you have the picture of the mushroom in your head. So you know what kind of fungus you are describing. So this is another Lepiota species based on both macro and micro morphology. So when identifying mushrooms, it is important to provide as much as details possible. So we know in the MycoAsia, this mushroom identification page, people post mushrooms and ask what is the name of this mushroom. So what is the edibility of this edibility status of this mushroom, such things people often ask. But if you don't provide enough details, it's very hard to give a proper identification. And also, it is not good to give the names only based on few identification characteristics, because if somebody is asking because he or she want to eat the mushroom, then you are putting a person into risk. So if you don't get enough information, don't give the identification because it is very risky. So you need to give all the characteristics such as the top picture and the bottom picture, and what kind of trees are associated with this mushroom, and also whether there are any color changes when you cut it, and what is the elevation, or the whether it is from a tropical area or temperate area. All this information, if you give all this information, is very easy for a person to give the identification. So when you dry a mushroom, please remember dry it below 50 degrees, because if you use temperature above 50, this can degrade the DNA. So later when you extract DNA from dry mushrooms, you don't get a good harvest of DNA. So please remember to dry your mushroom below 50. And once you dried, you store your mushrooms in plastic bags. And also it is important to add some silica gel in case if there are some moisture inside, so this silica gel help to eliminate those. Otherwise, the mushroom can be contaminated by other microfungi. So then you cannot do anything later on. And here I am going to show some common genera, how they are identified, and then often people confuse these genera with some other mushrooms. So one is Wolvariella and Amanita. So both these mushrooms look very similar, but these can be separated based on these characteristics. So if you see Wolvariella, these gills start with white when they are young, but when they are mature, it becomes pink. So Amanita gills remain white, so they don't change the color. It means Amanita has white gills, but Wolvariella has pink gills. And the ring. So some Amanita species, not all, has a ring, but Wolvariella does not have a ring in any species. And volva, so both has cup-like volva, but in some Amanita, you don't see a prominent volva like here. But Wolvariella always has a volva at the base. So if you see the growth habitat, so Wolvariella often grows on wood chips, rich soil or compost. But Amanita, you can see they always grow associated with ectomycorrhizal trees, oak or conifers, such trees. So these are the characteristics that you can identify these two apart. This is very important because Wolvariella is known for an edible mushroom, but many Amanita species are poisonous or some are deadly poisonous. So it is very important that you identify these two properly. And then the Wolvariella and Pluteus. So these two are also confusing. You can see Wolvariella has pink gills, but Pluteus also has pink gills. But Wolvariella has a volva at the base, but Pluteus don't have. So from this character, you can identify these two apart because Wolvariella has a volva, but Pluteus lacks a volva. And then chlorophyllum and macrolepiota. So this is also a confusing two genera. So chlorophyllum has olive green spore print or olive green spores. But when they are young, you don't see this olive green. When they are young, you see it is white. So how do you identify this? So one is macrolepiota. You can see the stipe has this like the snake skin pattern on the stipe, but you don't see it in chlorophyllum. Chlorophyllum has a very like pale, not like macrolepiota stem. And if you can find some mature specimens, you always see this olive green gills or the spore print. So this is very important because chlorophyllum is a poisonous species, Chlorophyllum molybdidis, but Macrolepiota procera, it's a edible one. So it is important you identify these two correctly. And then the next confusing one is Agaricus and Lepiota. They both also have like similar characteristics of their cap. So, but these two can be separated based on also the gills. So Agaricus also, when they are very young, it is white and then turn into pink and then finally turn into chocolate brown. But Lepiota, always white. And when you see an Agaricus, if you are confusing because it is, when it is young, it has white gills, but you can try to find a mature one. You can see like the gills a little bit pink or chocolate brown. And Lepiota and Cortinarius. So, Lepista and Cortinarius. So Lepista is a good edible one, Lepista nuda, and Cortinarius, some species are poisonous. So very clear distinguishing characteristic is this cortina. So cortina is like a spider web under the cap. So when the spores fall into this web, you can see it is like rusty brown things on a spider web. So if you see this under the cap, so this is definitely a Cortinarius. But in Lepista, you don't see any ring-like structure or spider web-like structure. So you can easily separate these two apart. So now I am going to show some tropical genera that are very common. So these are Termitomyces. So one of the important things that you can identify Termitomyces, when you touch the top of the mushroom here, you can see like a notch. So this is because Termitomyces grow inside the termite nest. So when they want to come up, so they need a sharp point at the tip. So you can always see like this notch on the top of the mushroom. So by using this characteristic, you can easily identify Termitomyces. And this is another Termitomyces species. And this is also, I see very often, like people post and ask, what is this mushroom, whether this is edible. This is Lentinus squarulosus. And this is very characteristic mushroom, grow on dead wood and has scales, very characteristic scales on the cap. So this is a good edible one. Yeah, this is also Lentinus squarulosus. And the Volvorella I mentioned earlier. So you can see the very clear volva, and the cap characteristic is totally different from Amanita. So Amanita always has like very shiny and sometimes like sticky cap, but you don't see that in Volvorella. And Pleurotus giganteus, also a very good edible one. You can see this characteristic pattern on the stipe, on the cap, so you can easily identify this mushroom. This is also a very good edible, very common in tropical areas. Yeah, this is also Pleurotus giganteus. You can see this characteristic pattern on the cap surface. And the Lentinus conatus, also an edible one. And so it is better you harvest these in young stage, otherwise it is very hard So it is like rubber when you cook it. And this is Lentinus sarchakaju. This is the only Lentinus that has a ring. All the other Lentinus, you don't see a ring, but this one has a ring. This is also a good edible one. And phallus, earlier this was in separate genus, Dictyophora, but now both the ringless and ringed with the netless and with the net, both genera merge into one genus, phallus. And marasmius, you can see always grow on dead wood or dead leaves. Cortinarius, as I mentioned, so cortina, it's a very important characteristic to identify. And Coprinellus disseminatus, very common in tropical areas. Before it was Coprinus disseminatus, but now changed into Coprinellus disseminatus. And Lentinula, also a very good edible one. Silaria is a Ascomycete. And Auricularia, very common in tropical home gardens, and edible one. Yeah, this is also Auricularia. Promella, it's a very good medicinal mushroom, grow on dead wood. Ramaria, Clavicorona. This is Clavicorona pixidata, also grow on dead wood. Psilocybe, or the magic mushroom. So psilocybe has bluish black spore print. And when you bruise the cap or stipe surface, you can see it goes blue. Depending on how quick this goes blue, indicates how strong this magic mushroom is. Like if the mushroom go blue very quickly, that means that has a high concentration of psilocybin or psilocin. Yeah, this is the underside. Goes the spore print is like blue to black. And another psilocybe and Paniolus, also one of the psilocybin containing groups. Lycoperdon, another species of Lycoperdon pipefarme. And Cythus, the bird nest, also another bird nest. And Philobelitis manipularis. So this is a bioluminescent mushroom. So when you see this in tropical forest at night, so you can see this emits a white light. This is also Philobelitis manipularis. And Flavolaceae, also an invasive species, spread very fast. Can see in many places. Flavolaceae calocera. This is also another Flavolaceae. Ophiocordyceps, or the entomopathogenic fungus. This is Termitomyces, the smaller one. So when you see this smaller Termitomyces in one place, you can see they appear in several places. This is because the termites, they cultivate the smaller ones in different places away from the main termite nest. And Amanita, this is Amanita hemibaba, which is an edible species. Phallus or mutinus. And this is the egg stage of a phallus species. When it cuts open, you can see the structure like this. And Azaro rubra, this is also common in tropical areas. And Clathrus delicata, it's a very small basidiomycete fungus. And Lactarius volumus is an edible one. So Lactarius, you can always identify by checking whether this has milk. When you break a little bit, you can see the milk comes out. And Rasula, Ganoderma, also a common one in tropical areas and also medicinal. And Phyloporus. So this is the only Bolitaceae member that has gills. Normally Bolitaceae, they have spores, but this genus, Phyloporus, has gills. Agaricus, also Agaricus, you can see this in this stage, the spores are pink. Entaloma, Mycena and Rhoridomyces. So you can see Rhoridomyces always has this kind of gelatinous structure in the stipe. This is Mycena longicide. You can see the hairs on the cap. Odumancella, Zerula, and Hygrocebi. It's a colorful mushroom. Hygrocebi roseoflava. And the Lepiota, also another Lepiota. Inocibi. So Inocibi always has this kind of fibrous cap surface. And the stipe base is a little bit swollen. So you can identify this genus based on these characteristics. In Pleurotus, Sathirella. Yeah, Sathirella is very common in tropical areas. Microceliota is very closely related to Agaricus. And Microceliota, when they are young, you can see the gills are white, but when they mature, the gills become a little bit like light brown, but not become chocolate brown like Agaricus. So you can identify the two genera apart. And most Microceliota are smaller, but most Agaricus are huge mushrooms. And Cantharellus, a very characteristic structure below the cap. Yeah, this is Cantharellus cibarius. And Fistulina, also a good edible one, Fistulina hepatica. We call beefsteak fungus. Yeah, this is Fistulina hepatica, also the same. And Macrolepiota procera, Clavaria, and Clavulina. Colibia always grow associated with the leaf debris. Cletopilus has a unique smell of farinaceous smell, or the flower smell. So you can recognize this genus by this smell, characteristic smell. Also Cletopilus. Geastron, or the earth star, and Leucocoprinus delicatissimus, and Pseudohydnum. This is like hydnum. You can see the tooth-like structures, but it is called Pseudohydnum because they are not the true hydnum type mushroom. And Stropharia, Trogia, and Crepidotus, and Wolveriella, as I mentioned. So these are the main tropical genera that you see mostly in the field. So I'm not going into very much detail, but I believe I mentioned what are the most important characteristics that you need to consider when you identify mushrooms. So this is my take-home message. So identifying mushroom is not an easy task. So incorrect identification can lead to deadly results. As you know, some are deadly poisonous and some are good edibles. And also remember, mushroom identification is not rocket science. It's also not very difficult. But if you need to have patience and you need to have dedication, patience and everything. So then you get to know your mushrooms. So that's all. Thank you very much. Again, Dr. Shanoy for inviting me to deliver this talk. So thank you. And if you have questions, now it's time.
Thank you, Sam. I hope you can hear me.
Yes, I can hear you.
Thank you for that masterclass on mushroom identification. You may please stop presenting so that we can get a better view of you.
Okay.
No, not that. Not that.
Okay.
Stop. Sorry, Sam. There was some confusion. I think I'll let you stop. Wait, please. Can you unmute yourself, Sam?
Okay. Sorry. I clicked
No problem. No problem. In the meantime, we got some many interesting questions. Actually, maybe we can start answering that. Why the chemical tests are necessary for field identification, Sam?
Sorry. Could you please repeat the question?
Sneha wants to know why the chemical tests are necessary for field identification.
Chemical test, right?
Yes.
Yeah. So, as you all know, there is no universal test to identify mushrooms, whether it is poisonous or edible. But we have certain tests that have been developed to identify certain groups of mushrooms, whether these groups, some are edible or some are poisonous. I can say an example. For example, agaricus, we have one chemical test called Schaeffer's reaction. So when you do this Schaeffer's reaction on agaricus species, so you use concentrated nitric and phenol. So you cross, you draw one line with the phenol on the cap of the mushroom, and then you cross that line with the concentrated nitric. So in the cross point, if you get a red reaction, so that means that is Schaeffer positive. That means it is edible. But if you don't get this Schaeffer's positive reaction, that means it's not edible. So this is one test that has been developed to specifically identify some groups in agaricals. So like this, we have certain tests for different groups. So this is very important for identifying certain groups of mushrooms, not all the mushrooms.
Thank you, Sam. The next question is, what is the best sample? I hope you can hear me, Sam.
Yeah, I can hear you.
So what is the best sample for DNA extraction and sequencing, whether the dry sample or the fresh sample?
It is always the best is the fresh one. So also, if you can get a culture, because for cultivation purposes, sometimes we isolate the mushrooms into a culture. So in this case, you can use the culture of that particular mushroom to extract the DNA. So you can get a very good harvest of the mushroom. Or if you don't have the culture, use the fresh mushroom. You can get a very good harvest. But some cases, you don't have the fresh mushroom for DNA extraction. So you can use the dry one. But as I mentioned, you should dry it below 50 degrees. Or you can put a small piece into a vial or a small Ziploc bag and put silica gel. So you can keep it dry and can you use it for DNA extraction.
Thank you, Sam. The next question is from Dr. Mohammed Javed. He wants to know, do you have any experience about how to culture these mushrooms?
Yeah, culturing mushroom, as I mentioned, so we can culture the mushrooms, the saphrobic mushrooms, using PDA. PDA is the universal medium. So you take the mushroom and tear it open, the cap inside a laminar flow, and then you take a small sterile piece from the tissue and dip it in the PDA plate, and then you put it in the incubator. Then you can get a nice culture.
I think the whole mushroom industry is based on the culturing of mushrooms, right?
Yeah, I mean, you need a culture to do small scale experiments first, and then you can scale up later.
Thank you, Sam. Next question is from Mr. Karthik. His question is, can you please share some resources like books, website that help us in mushroom identification?
Yeah, this one I can send to Dr. Shanoy, and then maybe Dr. Shanoy can share with him or everybody who wants this.
Yes, thank you, Sam. I'm sharing my email address. They can contact me and I will connect. I'll share the resources once you give them to me. Thank you.
Yeah. So this is, sorry, Dr. Shanoy, this is a big problem in tropical mushroom identification because we don't have proper guides like in Europe or in the US. They have like good guidebooks, but in tropical areas, we still have a lot of troubles identifying mushrooms. So, but I will give like as much as resources for them to read and get the knowledge on identification of tropical mushrooms.
Professor Sam, tropical mycology has been happening for a long time now. Why are we not having our own guides?
That one, I don't know. I mean, because for a guidebook, to have such a good guidebook, you need to dedicate yourself and you need to spend a lot of time. Maybe because the tropical mycology start to boom not too long ago. Because in Europe, in the US, they have been doing mycology for a long time than the tropical mycologists. So I think in the future we will have some guidebooks, because now start booming. Especially Dr. Shano is trying to promote the tropical mycology. So I really appreciate it. And we hope in the future we will have some good guidebooks in the tropics.
Sam, I have been getting similar frequently asked questions. What is the name of this mushroom and can I eat it? So then I need to contact you. So better to have one platform where we can share all the relevant information, I guess.
Yeah, at least we need a good platform for these common edible ones. So then people won't get poisoned.
I think we will think about it. Thank you, Sam. Next question is from Frank Rodriguez. When doing the collecting, would rubber gloves suffice against possible toxic mushrooms that might be encountered while collecting, or are there better durable materials for gloves? If not, are they that much scary, the toxic ones?
Touching will not do any harm, so you don't need to worry. But please remember, when you, after you done your collection, always wash your hands with like soap or something to remove all the mushroom remnants in your hands, because sometimes maybe you eat something in the field without washing your hands, so this may cause some problems. So to avoid this, always try to wash your hands once you are done with the collections. It is totally fine. You collect mushrooms with your bare hands, no problem.
Next question is, if we are working on dried mushrooms, how do you suggest that we rehydrate them for laboratory observation purposes? Is soaking them in water enough?
When we observe for micro characteristic observation, we use 5% KOH. This is the best to rehydrate the structures. So you section the mushroom and then put it on the slide and then put a drop of 5% KOH, and then you cover with the cover slip. So this will perfectly rehydrate the structures in the mushroom.
Thank you. So the next question, I would like to prefix it with my own question. Is it really necessary to look into morphology when it comes to mushroom identification to the species level? So the original question is, I would like to ask whether molecular identification of a fungal species is always necessary.
Yeah, as I mentioned, the primary characteristic should be the morphology. So you can use phylogeny as the secondary method to support your morphology, because nowadays without phylogeny, no journal accept the papers. That is the reality. So you need to incorporate both morphology and the phylogeny together, because after you done your macro and micro morphology, so you can see how phylogeny support your morphological identification. So it is necessary. Like I would say, it is necessary. But as you can see now, people describe new species based only on phylogeny. So this is, for my opinion, I don't agree. Like as I mentioned, how can you imagine a mushroom by looking at a sequence? When you do macro and micro morphology, you have the image of the mushroom in your head. And sometimes people ask whether it is necessary to draw the micro characteristics of mushrooms. So for me, if I draw the characteristics, I can better remember the mushroom. But I know nowadays you can take photos of Basidia, Cystidia, and all these structures. You can take photos and publish. But if you want to remember your mushroom well, try to draw it. I know it takes time, but at least you can do it and see yourself.
Sam, I beg to differ because looking at the psyche of the current generation and the way the fungal taxonomy is progressing with whole genome data, omics data, I am visualizing fungal taxonomy more of database. I mean more of data centric, less about appreciating the morphology. You can use morphology probably for aesthetic purposes. Oh, it looks beautiful, blah 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 blah. But when it comes to classifying, you would probably rely more on other data. When we talk about polyphasic, it doesn't have to be morphology first. I always try to visualize that way. Anyway, thank you for your input on that. Yeah, thank you. Next question is, I'd like to know, somebody wants to ask, one of the audio, participants wants to know whether we have already asked, if we think mushroom found in the field may be rare, should we pluck it for identification, sir?
Sorry, could you please repeat?
If you find a mushroom in the field which may be rare, should we pluck it for identification, or what should we do?
So, in the field, when you find a mushroom, in the first place, how can you know that it is a rare mushroom or not? Like, so you need to do a scientific identification in order to know whether this is a rare mushroom or not. So this is what happens in most of the countries. For example, in Sri Lanka. So people always say, like, our biodiversity is very high. We have a very high diversity of fungi, mushrooms. But nobody has done any survey to find out what we have and what we already lost. Like, so the first thing we need to do a proper survey to understand what mushrooms or fungi we have. And maybe some mushrooms or fungi have been disappeared without knowing. So whether it is rare or not, we cannot judge it in the field. We need to collect it and do a proper identification in order to know whether this is what kind of mushroom. So we actually need to collect it and do the proper identification before coming into any conclusion.
I think that was coming from an experienced mushroom forage or mushroom enthusiast or expert. Basically, he wants to know, or he or she wants to know, I know this is rare, but should I pluck it for the further studies, or should I leave it in the nature?
Yeah, I mean, even you pick it, it doesn't harm because the mycelium already inside the substrate or the place that the mushroom was growing. So even you pluck it, it doesn't harm. You don't destroy the whole the population. So it doesn't matter. You can pick it, but you can, when you see like several fruiting bodies, try to leave at least 1 or 2 fruiting bodies so that the mushroom later can disperse the spores and spread the generation. So that is totally fine. You pick the mushroom.
Okay, thank you, Sam. Somebody asked a very interesting question. I would like to know whether it is necessary to taste the mushroom during field for identification.
Yeah, this is what I mentioned. Like, if you know the genus is Amanita, or like known for deadly poisonous, I suggest don't do it. So if you know it is a normal mushroom that is known like lentinus, pleurotus, this kind of mushroom. So you can do a test. As I mentioned, you can take a small tissue and put it in the edge of the tongue and see whether like what kind of feeling you get, like burning or spicy or what. But don't try for these kind of deadly genera.
Okay, thank you. So next question is, do you have time, Sam? Maybe 10-15 minutes more?
Yeah, sure.
I mean, because we have got some really nice questions here, actually. Sana from Pakistan wants to know, how can we decide whether unusual morphological traits are natural variations or indicators of new species? I want to add that. When I was doing my Ph.D. in Hong Kong, the unofficial guideline was that if you find two characters different, then you can describe it as a new species. So is the rule still valid, and what is it now?
Yeah, so there is no like cutoff. Like, for example, in phylogeny, like how many base pair difference you can describe as a new species. We don't have a clear definition. Like, okay, 5 base pair difference you can describe a new species. Okay, based on the morphology. So morphology, as I mentioned, like if you have 2 collections of a mushroom, then you really know like the characteristics that you are seeing are not because of mutation or something else. So that's why it is important you have 2 collections. But in morphology also, you need to have like enough like evidence to put them apart. Like nowadays I see, like especially in microfungi, like people are describing so many new species based solely on phylogeny, like this much base pair difference, okay, described as a new species. But I think for me, I suggest at least check like morphological differences and also phylogenetic differences, and also, as you said, polyphasic approach, like the chemical profile and also other secondary metabolites that they produce, and the ecological differences. Consider all these and then try to give a fair identification, not just based on 1 or 2 differences in morphology, and then you describe a new species.
I think the question pertains more about this species delimitation. Then I would like to share my experience with Coltrotrachum siamens, and I have been talking about it for almost a decade now. Probably people are bored of it. But thing is that you can test whether these individuals belong to one species or multiple species. You can test their genetic barrier. You can prove their genetic barrier. There are methods like you can use the gene sequences. So I mean DNA sequences from 6 to 8 gene regions. Then you can use coalescent methods, and based on the coalescent methods, you can tell whether the recombination is there or the genetic barrier is quite strong. If there is a genetic barrier, that means they belong to one species. So there are tools more than in addition to morphological observations. I think that would need serious consideration. Thank you. Next question. So Akila has many questions. about specific, like there are some specific questions. I don't want to delve into them. Blah, 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 blah. These are very specific questions about two genera that I'll ask her to contact you, Sam. Okay. When Sana wants to know when a morphology and molecular data give conflicting signals, how do we decide which evidence should have more weight in species identification? Sam?
Yeah. So in cryptic species, so we always get this sometimes the morphology is similar, but the phylogeny is different. So sometimes the morphology is different, phylogeny is similar. So if you apply a polyphasic approach, so then you may find some differences. Like here you talk only about morphology and phylogeny, but if you apply other aspects like the chemical profiles and the ecology and the other aspects, then maybe you find some differences. But as I said earlier, so if you don't find enough evidence, so just leave it as a non-species, like based on the comparisons, or leave it until you find more evidence to describe as a new species. But I see some people more evidence. So this is a problem.
We have one more interesting question, Sam. What is the easiest and safest way for local people to identify mushrooms at home?
So as I said, there is no easy, like shortcuts. So you need to observe like mushrooms very carefully, like the color of the gills, the characteristics of the caps type, and these things, because sometimes people are very excited when they see like mushrooms in the field. I saw in Assam also there were several mushroom poisoning cases. I imagine they confuse with the lookalikes. So there are not shortcuts. So you need to like carefully observe and understand whether you are trying to eat the proper one or the wrong one.
Thank you, Sam. The last question is from Sana. Sneha Majumdar, I think. Sneha, she wants to know, unlike plant taxonomy, is it necessary we should do mycological illustration? Please suggest the proper channel, probably platform or resource where we can learn how to do. Sam.
Yeah, so it is not necessary, as I said. Now there are like techniques that you can take good photographs of micro characteristics, but it is good if you can learn it and try it at least once, so you understand like the difference between the fast technique and the traditional like drawing technique. So for me, I can remember the structures and everything properly when I draw it, because when draw you need to move the slide and see different shapes, different sizes, and like you observe many spores and you understand like how this varies. But when you take photographs, you want to do it quickly, so you take photographs and make a photo plate and do it. So that's it. But if she needs the details how to draw mushrooms, I can share with Dr. Shanoy and you can share with her.
Thank you, Sam. Thank you very much, Professor Samantha Karunarathna. for sharing your knowledge with the community. I mean, that is very important. And probably this has been one of the best sessions so far on the mushroom identification. And the credit goes to the expert who has delivered the talk and interacted with the audience. Credit also goes to the participants, today's participation. I mean, we have around 30 people here today. And the kind of questions we had, I don't think even if we had 200 participants, we would have got that kind of. Previously, we used to get 100, 200 people in the audience. But with this small audience of 30, 35 people, the kind of questions, serious questions and probing, I don't think we have done it so far. So it has been a great learning experience for me, and I'm sure this has been a similar experience for the other participants in today's session. I once again thank Professor Samantha Karunarathna for sharing this knowledge and being with us today. And thank you very much today's participants. You have been awesome. You have been wonderful. You have been quite appreciative of the whole process of learning today. And with this, we sign off. The video will be on YouTube. People are free to revisit the recordings and learn more. And probably you can be in touch with Professor Samantha on Facebook. And of course, and the Fungi Planet group also. You can Google him and you can find out his email address. Feel free to contact him. And we congratulate Professor Samantha for becoming the editor-in-chief of new fungal taxonomy journal, Fungal Taxa, Systematics and Evolution. And we hope to have a great time with that journal also. And we will have more, we'll have, I think, webinars under that platform too. Thank you so much, Sam. Thank you very much, ladies and gentlemen. Bye-bye.
Yeah. Thank you very much, Dr. Shanwai, and thank you very much all the participants for such an interesting questions. And thank you all.
Yeah, thank you. Take care, everybody.